as one of the people who've been around since the very first discussions of lung map, I just have to compliment you and the entire team. It is amazing to think how much we have learned from your experience, and we know how hard it's been. So let's move on now, and we're going to hear from Rajeshwari Sridhara from the FDA.
Thank you. I want to start with, again, remembering Dr. Dan Sargent, who was a dear friend of ours, and Shilsky started this morning recognizing his contributions, and he has done a lot in clinical trial designs. So, just for the purpose of making sure that everybody is on the same lines here, that adaptive design allows for plan design modifications. I know that Rich previously asked why it should be planned. Modifications based on data accrued in the trial up to the interim time, and unblinded or blinded interim results can be used. And specifically, we want to make sure that we are not looking at placebos, toxic placebos, and so false positive rate becomes important. And control of even operational bias. When you're starting the trial, you may not have everything set, and so we don't need to control operational bias. And it assumes that independent increments of information are coming in. So by definition, a study that includes a prospectively planned opportunity for modification, not that you have to necessarily modify, but it allows for an opportunity of one or more specified aspects of the study design and hypotheses based on analysis of data. That's interim data from subjects in the study itself. So there are of course, exploratory adaptive designs and hypothesis testing or confirmatory studies. In the exploratory space, there is less restriction. You can explore as much as you want without adjusting for multiple looks, multiple adaptations to generate hypotheses to be further tested. So there is a lot of leeway here when you're doing exploratory analysis, and some of the phase 1, phase 2 studies fall into this space. But as soon as you're testing hypothesis for a confirmatory reason, or you want to test the hypothesis itself, that one drug is better than the other, or that they're almost the same, then you have to think of adequate and well-controlled studies. And here you do need pre-planning so that you don't come up with a false positive conclusion. And decision rules specified for each adaptation. So here, I just want to go back to what Rich was asking about, why they should be pre-planned. So we are not saying that you should not have very many adaptations, but what is your threshold? When are you actually thinking of this adaptation? So that's important. It's not necessarily what result you are going to get, but you should have those thresholds set up front. So we have two broad kind of adaptive designs that we have been using. One is the group sequential design, which most of us are familiar with, and almost all cancer studies use this group sequential designs. And during the conduct of the study, data are analyzed and reviewed at periodic intervals, and these intervals are pre-specified. And here we assume that there is a correlated increment of information that is flowing in. So using interim estimates of treatment effect, you could either continue the trial or stop the trial, either for futility or actually efficacy. You can also decide to modify and say, you want to increase the sample size, or you can even say we are going to only study a subset beyond that interim analysis. The Bayesian design is a different paradigm where there is no actually hypothesis testing at all. Here we assume that the parameter measuring the treatment effect, like the hazard ratio or any of those, is regarded as a random variable, and the Bayesian inference is based on the posterior distribution, or what is the posterior probability that you will get a certain treatment effect based on what you have observed That's what you're concluding there, and not a hypothesis test. So then we, some of the things Mary talked about, enrichment designs, where you enrich the study population. And enrichment could be by prognostic markers, and this we do very well. And we have always used the first-line treatment, second-line treatment, etc. They're all different types of enrichment that we are doing, and this depends on the risk of the population. And whether the events occur in a shorter period of time or a longer period of time, or triple-negative metastatic breast cancer, or specific histology, etc. So these are all forms of enrichment, but they are based on prognostic markers. When we are talking about enrichment in general, we talk about predictive markers. Here we have, of course, examples of Herceptin for the HER2 positive breast cancer, as well as vemurafenib for melanoma with BRAF mutation. So these are specific predictive markers. What we mean here is that either there is no excuse me, there is no effect in the negative population, or that the effect in the negative population is much smaller, and therefore we are looking at only the positive group. So then you could have adaptive enrichment trials where you start with all comers, but at interim analysis you see that there is no effect in the negative, or whatever your threshold is for that. And then you can stop accrual in the negative population and only accrue further in the marker positive population. Master protocols, Mary touched on this. Here you have an overarching protocol that includes one or more of the following: multiple diseases, multiple treatments, multiple molecular biomarkers. So basically here you're trying to answer multiple questions, or you have multiple objectives. So you may have one treatment with multiple diseases, multiple treatments for one disease, or you may have both multiple diseases and multiple treatments. And these have been given different names: platform trials, umbrella trials, basket trials, but they are all essentially master protocols. So again, the characteristics are: this is a one trial, there is a central governance structure, and central IRB, central DMC, central independent review committee. These are all good to have, but not all of them may have. And you're studying multiple drugs, multiple markers, and you can leverage a common control. And there is a flexibility in these master protocols to add or remove agents, or even add an extra study into it, as in the master protocol that Mary was presenting in the lung map. But in the same space of master protocols, you can have non-comparative studies. The very first one, which was not named as a basket trial or a master protocol, was the imatinib trial, where there were I believe 40 plus diseases that were studied in that study. But then imatinib was approved by earlier approvals, and it had a proven efficacy and safety profile, and 5 specific cohorts from that group. These were all single-arm cohorts. It was just a cohort of multiple diseases, all of them treated with Gleevec. And they all had the common molecular biomarker. And the drug approval was based on response rate from this study. On the other hand, NCI match, which is going on, is an exploratory study. It's very important here, too, that these exploratory studies are needed to establish where a product may work with what molecular marker subgroup, and the treatments are matched to different markers. So again, here you have, it's not defined by histology, but multiple molecular markers and multiple treatments that are being studied under one protocol. And we have had these exploratory comparative studies as well. And these have used Bayesian response adaptive randomization. So here, the randomization scheme itself changes as the data accumulates. And the battle 1 in advanced non-small cell lung cancer, and I-SPY 2, which I'm sure many of you know, are the two well-publicized clinical trials in this space. So again, in the I-SPY 2, for example, there was a control, whether it was, so they divided broadly into HER2 positive or negative, and within them, there were markers that were being studied, and you could, based on that, they were being matched as they got more and more data. So this was an adaptive randomization itself based on the responses seen within the study. These designs get complicated, and therefore the original objective of how can you show that a drug actually is superior to what you have or about the same gets into too many weeds, and so it's difficult to estimate an unbiased estimate of the treatment effect itself, because it's a biased randomization that you're doing based on the results. So this was talked quite a bit in the morning about the phase 1-2 expansion cohort studies. So you start with a dose escalation study in all solid tumors or hematological malignancies, and amend protocol to start expansion cohorts in specific diseases maybe, or with different dosing regimens. It could be single-arm randomized studies. Again, these all fall into the idea of answering multiple questions under one protocol. And again, it can be central governments may be coming from one individual industry or sponsor, or it may be coming from outside as well. So things to consider in these type of studies are you have to have pre-specified starting and stopping criteria. What is the maximum sample size needed? At any point you're expanding, what is your objective? Remember that these are still first-in-human studies which has been expanded to all these cohorts. So patient protection is of most importance in these studies, and exposing patients to unknown safety risk is what is at stake. So for these expansions, there has to be a threshold set up, a pre-planned adaptation of when are you going to the next expanded cohort. And it becomes difficult in data tracking and data dissemination. When you have multiple cohorts that are there, then the safety coming from one cohort, is it being disseminated to the other? What you're learning from one disease, is it going to the other, especially if the investigators are very different in the two disease areas? So I think this you already saw. So a phase 1 trial with over 1,000 patients, yes, there was a reason for doing this, but it is not for all products, and one should really be cautious in making sure that the safety is not compromised. And the other thing is these master protocols are good, and probably we can do a lot more with common control arms. So you have several treatments compared to the same protocol in a given disease. And compare each treatment with the control. So you have treatment 1 compared to the control, treatment 2 compared to the control. There is no need for comparing treatment 1 to 2. Our regulations say that you have to be better than, it has to be effective. There is no comparative efficacy regulatory standards. So you don't have to compare to T1 to T2. And so here you can have equal randomization of C to T, T1 to T2, etc. And I think this will become clear with this example. If you have a hypothetical example to illustrate the gain that you may get with common control, let's assume that we want to see that there is a 50% improvement. So you have a hazard ratio of 0.5 with respect to overall survival. And let's assume that the median, this is a refractory disease, and the median survival is about 6 months in the control arm. And if you have 2 independent randomized studies, then you will need 336 patients. Whereas if you do a common control, then you can reduce this number of subjects by at least a third. So here is an example where we have how we can do resource saving and do more efficiently. Currently, there are 5 concurrent ongoing studies in advanced renal cell carcinoma. In each of these 5 studies, the control arm is sunitinib. And they're all asking the same, it's in the same population. And when you see this, and they're all going to be read out around the same time. It's not that one is very much ahead and the other is coming later on. A good place where patients could have been saved. You're doing 5 times the control. This could have been set up with a common control. So the one thing that I didn't put on my, I'm almost at this, my last slide, is with eligibility criteria. This was brought up in the morning as well. Can we expand this? Now, the idea of developing the product, whenever we are doing, we want a pristine trial where you have everything homogeneous, so you can see if there is an effect of the drug, you can see that in that trial. So by expanding the eligibility criteria or putting more high-risk patients, you will be making that heterogeneous, and if there is a moderate or less than what you would like, the treatment effect, then it cannot be seen in a heterogeneous group. And so that's the reason of doing these homogeneous group. But there is nothing written in designs that you cannot include, for example, performance status 2 patients, or pediatric patients, or geriatric patients. These are often excluded, and you can always put them in the studies. And we can have designs similar to what Mary presented. You have this subgroup, or it's a bigger subgroup perhaps, of this homogeneous group as your primary analysis, but the rest of them also included in the trial, and you can use that data as well. So there are ways of doing innovative designs, and we can certainly save a lot of the patients as well. And the other one is comparison of dynamic treatment regimens. I think this is where we are heading as to which comes first and which comes later on. So here is an example of US intergroup study of R-CHOP versus CHOP. A 2-stage random assignment was made. In the first stage, it was the induction of R-CHOP versus CHOP. And then in the maintenance, it was rituximab versus observation in patients who achieve CR or PR. So obviously, this is a subset of patients who were in the first stage. And the primary endpoint was failure-free survival. The failure was defined as relapse, non-protocol treatment, or death. And we do have statistical methods to deal with these. A weighted Cox regression analysis was conducted to remove the bias from analyzing only a subset of patients who are treated in the second randomization. And this was approved, and this is in the product label. But again, in practice, patients are treated with sequential treatments. And the decision of the next treatment in sequence is specifically tailored to that patient. However, what we are trying to do is, for the population, can we come up with a sequence? Without re-randomization, it's difficult to estimate the treatment effect. So what we see in practice is very different. And when we are trying to say for a population, this is how the sequence should be, it's rather difficult. If the subsequent treatment is different from the first, then attribution of efficacy to a product is challenging. So this is a regulatory challenge. For the approval of the product needs to show that there is an effect. But then if you have subsequently another treatment, which is another new treatment, then and then you see a survival advantage or even PFS advantage, can you contribute that to the first drug or to the second drug? So statistical methods for certain scenarios are available to determine the overall efficacy with a treatment regimen. I think this is where we'll be moving further, in my opinion, that this kind of dynamic treatment regimes are the ones where we need to pay more attention and studies need to be done, and we have to come up with a regulatory pathway of dealing with how we can isolate the effect of each of these treatments. With that, I want to just say that we are a very big group in CEDAR. The Office of Biostatistics has 8 divisions. We are 230 plus statisticians. Most of us, very few are masters, most of us are PhD statisticians, and my division has 40 statisticians, and we completely support the Office of Oncology and Hematology Products. Thank you.